Alright, welcome back to part 3 of macromolecules. This is on primarily dealing with protein structure, and we're going into nucleic acids at the end of this. So you guys wanted a quick review on protein structure, so let's get started here. The first type of protein structure we talked about was a primary structure, and remember, it's just the chain of amino acids linked together by peptide bonds. Can be a few, 25 to 30 amino acids long, all the way up to thousands of amino acids long. And remember, it uses those 20 main amino acids, and they're all linked together to form this primary structure. The secondary structure is you have these two formations of that amino acid chain. You have the beta pleated sheets and the alpha helix. Now, if you remember that these are actually occurred from hydrogen bonds forming between the backbone of those polypeptides, not that R group. And those the hydrogens and the oxygens form that hydrogen bond together, and that's what forms these different formations. And one of the cool things about this it talked about was that in your book it talks about that beta pleated sheets, all those hydrogen bonds in that, all these hydrogen bonds right in here, see them right here and here and here, form cause this, give it a lot of stability, a lot of strength. And it gives an example of a spider web, and the strength that a spider web has, even though it's very thin, it's very strong. It's due to those hydrogen bonds. The third type of conformation is tertiary, and this one's primarily with all the R groups, and there's lots of bonds that form between these R groups. The first one here is the hydrophobic interactions, and again, this is the one that kind of gets stuck into the middle of that protein, and all the other bonds kind of keep it away from any water molecules, because remember, it's hydrophobic. And then over here you have some hydrogen bonds and ionic bonds, and also really this is kind of a unique is this disulfide bridge, and it's just between the cysteine amino acids, because they both have sulfur in them, and it gives this kind of some stability and some strength in the middle part of that amino acid chain, that protein. So this is the tertiary structure. And then the last one is quaternary structure, and this is just kind of the overall structure of it, and it's the combining of different polypeptides to form that overall protein shape. A couple of examples that we have here are collagen, which is a triple helix, so it's 3 different polypeptides bonded together. And this hemoglobin protein here is actually 4 different, the book calls them globular proteins. 2 beta chains and 2 alpha chains come together to form this whole overall protein called hemoglobin that carries oxygen in your body. So that's a quick review on the protein structure. Let's jump into nucleic acids. Hopefully we can get all this done in enough time. Of course, you know nucleic acids as DNA and RNA. We talked a little bit about it in class already. But if you look at this here, it says amino acid sequence is determined by genes, which consist of DNA, which is a type of nucleic acid. Okay, what that's saying is all the proteins, okay, the proteins are composed of amino acids, and amino acids are basically determined by a blueprint of genes, and genes are just certain segments of your DNA make up a gene. So, and of course DNA, the N and the A in DNA stands for nucleic acid. So that's what the N and the A stands for. So that we're going to kind of go into this and talk about it a little bit in more detail, and we'll talk more about nucleic acids in further chapters down the road. This is kind of an introductory chapter on nucleic acids. So what is the role of nucleic acids? Now, I always use the analogy of blueprints. I think I may have mentioned this in class. If you're building a house, okay, if you're building a house, first thing you have to have before you build a house is you got to have blueprints to build the house. It's basically instructions for the builders to know how to build it. And that's kind of the way DNA is. DNA is these is a set of blueprints for your body to make proteins, which is basically what you're composed of. So in a nutshell here, if you look at it, the way this is written out, so DNA is found inside the nucleus of your cells, of all your cells, And just like with any, if you're building a house, you usually have like 1 or 2 sets of blueprints, but you might have several people that need to see a copy of those blueprints. You don't want to be given that. You want to have an original set. You don't want to be giving that original set out to everybody, all the different contractors. You don't want to give one to the plumber, to the electrician, to the framer. You don't want to give your original set. So what they will do is they'll make copies of that original set and they'll give it to all these different people to build your house. Same thing's going on here with DNA. And what happens here is that we have our original copy that stays in the nucleus. That DNA never leaves the nucleus. It's right here. All right, if you can see it, that's the DNA strand. And what happens here, your body will make a copy of it. All right, it will make a copy of that strand, and that copy that's made is a single-stranded RNA. And then that RNA is what will actually leave the nucleus, and it will come out here to this little structure here. It's called a ribosome. Okay, you'll learn about that in the next chapter. Ribosome is where protein synthesis takes place, and at that ribosome, well, that's pretty sloppy, isn't it? At that ribosome, the cell will actually make the protein. So that's kind of a quick review of DNA and how it works. So you kind of go from DNA to RNA and then to a protein. So that's kind of what that's saying there, DNA to RNA and then to the protein. A couple other things. Let's talk a little bit about the structure of nucleic acid, so you don't want you to get too confused about this. So we said the other day in class that over here, this is kind of shows you one side of a DNA molecule. And I kind of want you to think of that DNA molecule as like a ladder. So this is like one side of a ladder, and then you imagine there would be another side of it here. We'll talk about that in a second. But over here you have 2 parts to this side ladder. You have the phosphate groups and the pentose sugar, and you see them blown up in this picture. They make up the sides of that ladder, and then that nitrogenous base makes up the rungs of the ladder, okay, that sticks out here in the middle. Real quick, you'll notice here it says a 5' end and a 3' end. All that has to do with is which carbon is on the bottom, which carbon is on the top. So if you look, we always number our carbons with the longest chain of carbons. And so this happens to be carbon number 1 right here, carbon number 2 here. This is carbon number 3, therefore we call this the 3' end. If you look here, we start again, carbon number 1, number 2, number 3, number 4, and then number 5 is right there. That's why we call that the 5' end. And that's important, what we'll talk about here shortly about anti-parallel arrangement of the nucleic acids of the nucleotides. So another thing we'll look at over here, notice that the pyrimidines, you have cytosine, thymine, and I'm missing a U there. Should have a U right there. That's a uracil. Left the U off. I need to be more careful about where I find my pictures, I guess, don't I? And then the purines are adenine and guanine. So remember that adenine is always going to bond with the thymine, and the guanine is always going to bond with the cytosine. C's go with G's, A's go with T's. Now notice over here the uracil is only found in RNA, and it takes the place of the thymine, which is DNA. So if you're dealing with an RNA molecule, the adenine is going to bond with the uracil. If you're dealing with a DNA molecule, adenine is going to bond with thymine. So a little bit about that. I encourage you to look more about that in the book. We'll talk more about it in class if you have more questions about it. The next thing I want to talk about real quick is this double helix. This is a picture in your book, how DNA is a double helix molecule. And one of the things it talks about here in this part is DNA replication. So all these this is very important in that takes place in your cells. Before your cells will replicate and make more cells, they have the key thing. One of the key things that has to take place is that the DNA has to make a copy of itself, so that the new cells will have a copy of the DNA. And so what happens is really cool. This thing will just uncoil, kind of unzip, kind of like a zipper. It unzips, and this side of that DNA strand right here forms a template to make a new strand, and this side forms a template to make a new strand. And what you end up with are 2 new strands from that original strand, and they are identical to each other. They'll have the exact same nucleotide sequence. And we'll talk more about that later. We'll learn a lot more about DNA replication later, but just kind of want to introduce it to you there. And the last thing on this chapter is this anti-parallel arrangement. Earlier we mentioned that you have a 5' end and a 3' end. It all has to do with that carbon. Remember that carbon? Which end that carbon is on. Now, the way these things will run, they run what we call anti-parallel. So if you have this is the 5' end, when you look at the corresponding nucleotide sequence of that side, it's going to run in the opposite direction. Notice here you have an adenine, it's going to be bonded to a thymine. Down here you have a guanine bonded to a cytosine. So if you can imagine, this is one side. They kind of talk, the analogy in the book uses kind of like a highway running opposite of each other. So you're kind of running in this direction, and that's the way highways work. And that's kind of the way these are. So you got the 5' running this way, and then the 5' to 3' is going this way. 5 to 3, 5 to 3. Anti-parallel. Hopefully you understand that a little bit. We will talk more about that, especially when we get into DNA replication later on. So this is kind of a quick run through over DNA. If you have any questions, I encourage you to read your book, and we will talk more tomorrow in class, and hopefully be ready for the test very soon. So read your book, and you will be ready for the test. See you in class.